All right, so we're going to continue our discussion of T cells in the periphery. So far, we have seen the activation of a T cell via signal 1 and signal 2 by an antigen-presenting cell in a secondary lymphoid organ. We have seen the cytokine production, specifically IL-2 production, of that T cell as a result of its activation, and the autocrine or paracrine signaling of that IL-2 back to the T cell or its neighbors. We've seen the proliferation, and I've talked about the differentiation of those T cells, and we specifically talked about the differentiation and finally the effector function of the CD8 positive T cell. So it starts out as a naive CD8 positive T cell and becomes a CTL, a cytotoxic T lymphocyte, that has the killing functions that we saw last time. Today we're going to talk about what happens up here with the blue cells, and we're going to think about the differentiation and effector functions of CD4 positive T cells. When we think about differentiation and effector function of CD4 positive T cells, the cytokine signaling aspect of T cell activation becomes incredibly important. So we are going to see naive CD4 positive T cells become activated to induce their effector function. That process involves signal 1 through the T cell receptor, as well as signal 2 through a co-stimulatory molecule like CD28, but you're basically never going to hear me talk about them. Your textbook draws them in every figure, but we're not really going to ever focus on them today. Know that they are still happening. That whole business is happening. But we're going to spend a lot of our time focusing on this cytokine signaling step because it is so critically important for our CD4 positive T cells. It can be really hard to summarize CD4 positive T cells and their effector functions because there are many different subsets of those CD4 positive T cells. So when a naive T cell is activated, at the beginning it's just a naive T cell. It's not really one of these types of subsets. We'll get back to the one exception to that at the end today. But after stimulation, our naive T cell can become one of many different subtypes of T cells, each of which has a slightly different function. We can summarize all of these functions to say that all of them involve the CD4 positive T cell making some kind of cytokine that helps some other cell type. So these are our helper T cells. Once our naive CD4 cell is activated, it becomes a helper T cell or TH cell because it's helping. And the helping is always a matter of producing cytokines, but the specific cytokines will vary. I found this figure and this whole section of your textbook really interesting because it is so incredibly up to date. In fact, they talk about some subsets of T cells that I think are not completely accepted as subsets of T cells. I'm going to tell you kind of some specific requirements that we have for classifying a subset as a real subset. And one of the subsets that your textbook talks about, if you read through the paragraph on it really carefully, it's sort of like some people think, and this may be the way, and even that part is very, very tentative. And so I'm actually not going to talk about all of the subsets that your textbook talks about. I'm going to talk about sort of some that are particularly well established and well understood. There are a couple up here, specifically Th9 and Th22, that are less well understood. The Th22 section of your textbook is the one where they're like, well, maybe this happens a lot of times. So I'm not going to really focus a lot on those. All of our T cell subsets have in common, as I said before, that they are able to secrete large amounts of cytokine. And one thing that you should know about all of them is that just like with our CD8 positive T cells, this secretion process is directional secretion. So our CD4 positive T cell does not secrete cytokines in 360 directions to everything around it. Instead, our CD4 positive T cell is going to secrete cytokines directly towards a target cell, usually the antigen presenting cell, to specifically affect that cell. It's not as though the entire body is going to get affected. And in fact, the integrins on the side of the adhesion complex, at the peripheral supramolecular adhesion complex, or the PSMAC, do enclose those cytokines and push them towards the target cell, just like we saw with the cytotoxic molecules. So it is a directional secretion that's happening right here at the synapse between these two cells. This isn't a multi-directional thing. But the part of this where things get a little tricky is that our T cell, when it gets activated, will become one of those different subsets that I showed you on the previous slide based on which cytokine it receives at the time of activation. So our T cell is going to receive some polarizing cytokine at its time of activation that will push that cell towards a specific subset. I'm going to come back to this slide. I'm going to go to the next one and say a couple things, and then come back to this one and say a couple other things. So what really matters here is which cytokine the APC or sometimes other neighboring cells make to help that T cell when it gets turned on. That will dictate which subset that T cell becomes. The APC or the other surrounding cells, quote unquote, decide which cytokines to provide based on which PRRs got stimulated. So different PRRs could induce our antigen presenting cells to make different cytokines, and that will push the CD4 T cell towards making a different kind of response. Some of these different types of CD4 T cell responses are particularly good at getting rid of some type of pathogen. And so you could imagine a nice setup here where you have a PRR, say, that binds to a fungal product. Will lead to signaling in our antigen presenting cell, will lead to that antigen presenting cell making a specific cytokine. It will polarize our helper T cell in a specific way, and lo and behold, our helper T cell will become the kind that's really good at getting rid of fungal infection. And so we have this nice tailored response where a specific PRR, which you can see here on the left side of this slide, leads to signaling to make a specific cytokine that pushes our naive cell to a specific type of helper T cell. And so oftentimes people talk about the helper T cells as being something like the quarterback of the immune system, where they're deciding which direction to throw the immune system, which direction to push the immune system in for the subsequent response. If we were to use that metaphor, then the PRR would be sort of like the coach who tells the quarterback which direction they ought to be throwing in. In order to have this whole process work, our naive T cell will be exposed to and respond to a specific polarizing cytokine. And as a result of that response to the specific polarizing cytokine, this cell will turn on a unique transcription factor, or a master gene regulator. There's some transcription factor that is associated with each type of helper T cell. And the idea is that transcription factor will transcribe all of the genes that are important for that kind of T cell's job. And as a result, our T cell will produce a specific set of effector cytokines that will allow it to do its job. And so while people can sometimes come up with, the helper T-cell subset that responds to fungal infection in your left pinky on Tuesdays, like we can come up with lots of very specific types of T-cells, they only really qualify as a specific helper T-cell subset if we can identify the polarizing cytokine, a specific master regulator, a specific transcription factor, and a specific effector cytokine. And so we're going to see that for each of the subsets of helper T-cells that we talk about, and being able to name the polarizing cytokine, the master gene regulator, and an effector cytokine for each subset that we talk about is a key thing you should be able to name for each of them. Yes, Molly?
I know the answer is probably no, but is there any going back once differentiated into a specific subset?
So Molly wants to know whether the cells can go back. Once they've differentiated into one subset, can they go back? The answer is no, and there's actually even a little more detail to that answer, which you will see a little bit at the end of this. Okay? So the first people who started to realize that all helper T cells were not the same, that there were some different subsets of them, classified helper T cells into two subsets. They said there are two different kinds of helper T cells, or TH cells. And if you were the scientist at this point, and you had come up with this observation, what do you think you might call your cells? There's two kinds of TH cells. What would you call them? 1 and 2. You might call them TH1 and TH2. And in fact, that's what they were called. So our first two T-cell subsets were named the Th1 and Th2 T-cell subsets. And for a long time, there were only Th1 and Th2 T-cell subsets. I would say that the next one of these that was really solidly identified was identified during my first year of grad school. I was actually at the talk where it was first described, and I had no idea what I was listening to. I was like, what? But now I know. And so the Th1 and Th2 subsets were really the first ones that were well understood, and the ones around whom this whole paradigm was first described. So Th1 cells are the first of these important cell types. Your textbook goes through Th1 and Th2 cells, and only those two cell types in figure 10-11. Your textbook has a figure 10-9 that has all of the different subsets, and so I've basically broken out each one individually from 10-9. And your textbook also has a table, 10-3, that goes through all the specific subsets. So I'm just pulling out the unique details for each individual subset here. So Th1 cells are polarized specifically by a cytokine called IL-12. IL-12 is by far the most common polarizing cytokine for a Th1 cell. Yes, we can sometimes see some other cytokines like interferon gamma and IL-18 polarizing, but IL-12 is really the big one here. One other thing that I will say as I am talking through the T-cell subsets today, I am going to highlight particular cytokines, though you will see in some cases there are more cytokines than the ones I highlight that are shown on the slide. When I am grading exams, if it's one that's listed on the slide, you get it, it counts as being correct. Although, yes, in most cases, we do really focus on the one most important one. But yes, technically, you write any of those, you're right, as far as the exam is concerned. But IL-12 is the one I really like. IL-12 is the one that people really associate with Th1 cells. The master gene regulator, the transcription factor that is important in Th1 cells, is called TBAT. And Th1 cells predominantly make this cytokine interferon gamma. So Th1 cells are polarized by IL-12. IL-12 induces them to turn on TBAT. TBAT tells them to transcribe interferon gamma. So this is all well and good to know, but you also might wonder, well, what does this do for me? Why do I care that I have a T cell that makes interferon gamma? How does this help deal with some type of immune response? And interferon gamma has a number of different effects. As your book says here, it can enhance antigen-presenting cell activity and enhance T cell activation. The biggest thing that I think about is in the way that it protects against intracellular pathogens by activating macrophages. So I frequently think of a Th1 cell, that's been in my way all week, as activating macrophages and being really good at getting rid of intracellular bacteria. Once upon a time, we used to say just bacteria here, but now that we have more different subsets, some of the other ones are good for bacteria at other times. And so we can combat those types of organisms really well. So as I said, just to sort of clarify things, Th1 cells are really, really, really good at activating macrophages. So when a Th1 cell encounters a macrophage that has perhaps some microbes in its phagosome, that Th1 cell will produce interferon gamma, and that will induce the macrophage to do considerably more destruction of things in its phagosomes. So in fact, we're going to see more reactive oxygen species. We're going to see more enzymatic destruction in the phagosome. We're going to see way more killing of our bacteria that are intracellular. We are also going to activate that macrophage to have way more proteins that can stimulate T cells and stimulate immune responses on the surface. And so this is a key feature of a Th1 cell. One of the disease states where Th1 cells have been shown to be really important, and there are many of them, is in infection with Mycobacterium tuberculosis. Tuberculosis is an intracellular bacteria, typically, as you might be aware, found in the lung. And it is able to live inside of macrophages and, in fact, transform those macrophages into a nice, happy home. If you have tuberculosis, typically one of the things that if you've been exposed, even if you're not sick, even if you're not coughing and spreading tuberculosis to lots of people, sometimes you can still have the tuberculosis bacteria in your body. And the way that that's diagnosed is by a chest x-ray. On the chest x-ray, they're looking for something called granulomas. And in a granuloma, we have a bunch of Th1 cells, which are these little blue cells that are kind of washed out looking on this slide, that induce the activation of many, many, many macrophages. And those macrophages in this area actually form a wall around the infected macrophages, so that whenever those infected macrophages die, whenever they release bacteria, these released bacteria are walled off and are just going to get eaten up by new macrophages instead of potentially spreading out throughout the lung and causing damage. And so for many years, your body is able to actually contain that Mycobacterium tuberculosis in this walled off area called the granuloma, where we make a wall of macrophages. And in fact, we end up making a wall with some connective tissue as well to block that bacteria off. And this is all happening as a result of the Th1 response. One of the ways that we can show this importance is if we look at infection of mice with mycobacteria. This graph is showing infection of mice with a mycobacterium known as BCG, which is a relative of tuberculosis. It's not the one that causes tuberculosis. And it's somewhat less pathogenic. You can see that if you infect wild-type mice with BCG, they live up to 60 days, 100% live. If, however, you use mice that don't have interferon gamma and thus are unable to be making this Th1 response, you can see that those mice quickly die of their BCG infection. And so this Th1 response has been shown to be particularly important for the control of Mycobacterium tuberculosis. When Th1 cells were first identified, they're at that time opposite. Now there's a lot of subsets, so it's not like they're opposites anymore. Their other type of cell, Th2 cells, were also described. Th2 cells are polarized by IL-4. IL-4 induces these cells to turn on the transcription factor GATA3. And GATA3 induces the cell to make IL-4. If you look at this, you might find an area of confusion. Can you see anything that seems a little weird about Th2 cells? Yeah, Molly? It's turned on by the same cytokine it produces. And so you can kind of think of it like chicken and the egg problem. Okay, great, but where did the first IL-4 come from? If the IL-4 comes from the T cell, where did the IL-4 to make the T cell come from? And that's actually been a little bit of a point of contention in the field for a while. There are some people who think that this is actually done now by a cytokine called IL-33, while others still talk about IL-4. We know that it's actually a whole group of cytokines, IL-4, 5, and 13. They are right next to each other on the chromosome, and once one of them starts getting transcribed at a high level, all three of them do. And so Th2 cells are strongly making IL-4, IL-5, and IL-13. And Th2 cells are really important for protecting against extracellular parasites. And one of the big things that these cells can do is that they can activate a mast cell. I am drawing a poor version of a table on the board. So we've talked a little bit about extracellular parasites like this parasitic worm that is shown here. And one of the things that you should remember about things like that parasitic worm is that it's pretty big. It's not really going to be a size that your cells can very easily phagocytose. It is extracellular, as its name as an extracellular parasite. These Th2 cells can end up leading to a number of physiologic effects through the cytokines that they produce, IL-4, IL-5, and IL-13. One of them, as I listed on the board, is activation of mast cells and mast cell degranulation. One of them is eosinophil activation. One of them is a high level of antibody production, though we'll talk more about that later. And one of them is high levels of mucus secretion and peristalsis. And if you think about it, all of these are really great ways to potentially get rid of some extracellular parasite. If you have an extracellular parasite, you're not going to phagocytose it, but by using a mast cell or an eosinophil, you can degranulate, send toxic products towards that cell, and potentially lyse that cell with a toxic product. If you have a parasite in your GI tract, you want to expel all the stuff in your GI tract to try to get that parasite out. And so that's why we see things like the mucus secretion and the peristalsis. Remember that mast cells are particularly good at binding IgE. And so mast cells typically are coated with IgE, bound to their Fc epsilon receptors. When antigen comes around, it will bind to that IgE, and that will cross-link the mast cell and induce degranulation to potentially get rid of that parasite, say. A lot of this, of course, is happening with IgE, which is partially why we see a good amount of activation of this from Th2 cells, is that we can see some nice class switch from Th2 cells. And so this is sort of the general information or the general figure of the control of Th1 and Th2 cells. Our original dendritic cell gets infected with some type of microbe. In the top, it's shown as a virus. It could also be an intracellular bacterium. In the bottom, it's shown as a worm. That leads to the cell making different types of cytokines. In the top, IL-12. In the bottom, IL-4. The T cell on the top is getting a signal from MHC class II and from CD28, CD80 interaction. So we get signal 1 and signal 2, but we also get a signal through IL-12. That induces Tbat to come on. That turns the cell into a Th1 cell. That cell will now make interferon gamma, whereas at the bottom, our cell is going to make Th2 cytokines like IL-4 because GATA3 was turned on as a result of IL-4 exposure. These two also, especially in the beginning of our understanding of Th1 and Th2 cells, were also talked about in an interesting way because their cytokines that they produce will influence which antibody subtype is produced by an activated B cell. So when these different cell types encounter a B cell, they will induce class switch to different antibody isotypes. And again, this could help tailor the immune response to the particular type of pathogen that we are fighting. So IL-4 is going to induce class switch to IgG1 and IgE. That's how we're going to get that mast cell activation, because we turned on IgE, the one that goes with mast cells. IL-5 might turn on IgA production, so we're going to have something going on in the mucosal tissue. Whereas interferon gamma from our Th1 response will start to turn on IgG3 and IgG2A, which can be useful for different types of effector functions. So we also get these T cells biasing what happens with the B cells. I will admit that students sometimes find this sort of information confusing now. So when I first learned the T cell subsets, we had to learn all of this kind of stuff. As we've got, and especially we learned a lot about Th2 cells making IgE. As we've learned more about T cell subsets, there's one we're going to call the B cell one. And so then people are going to be like, but what? You already talked about B cells, what? And so realize that all of these CD4 T cells are going to be interacting with B cells in some way, pushing their responses in some way. But yes, one of them is going to be especially good at interacting with B cells. They all can interact and bias or polarize B cells in some way. But Molly asked a question, which was, can our cells go back? Once a cell has become a Th1 cell, can it change its mind and become a Th2 cell or something like that? And I told her that the answer was no. But a more important part of this answer is that not only do some of these cytokines and transcription factors actually induce differentiation into their own specific helper T cell subset, but they also inhibit the other. So IL-12 turns on Tbat. It's not really shown super well here, but IL-12 does inhibit GATA3. Tbat inhibits GATA3. GATA3 inhibits Tbat. IL-4 inhibits Tbat. So once a T cell starts down the Th1 path, it also begins really dramatically inhibiting the Th2 path. And so these are totally different outcomes and totally different paths for our cells. Further, the cytokines that are produced by Tbat are interferon gamma, and Tbat will inhibit IL-4 and IL-5 production. So we'll promote Th1 and inhibit Th2 at the same time. Similarly, GATA3 will turn on IL-4 and IL-5 while also inhibiting interferon gamma. because these cytokines can sort of feed back on the production of helper of Th1 and Th2 cells, sometimes we'll see a general overall response get biased towards a Th1 or Th2 response. A cell will become a Th1 cell, it will make interferon gamma, and that will help polarize its neighbors to being Th1 or Th2. So usually when you're making a response, you make Th1 in response to this antigen, or you make Th2. You don't tend to make a mixture 50-50 of each. These two subsets really inhibit one another. Sometimes that's a really good thing. Sometimes that's a really bad thing. So the first example I want to tell you about is an infection with a bacteria called Mycobacterium leprae. Mycobacterium leprae is the causative agent of leprosy. And it turns out that there are two different kinds of leprosy that you can see in people. Some people get the type that is shown on the left, which is known as tuberculoid leprosy. And in tuberculoid leprosy, we see relatively low numbers of bacteria. People with this type of leprosy are not able to infect others terribly well. They have granulomas, they have some local inflammation where they're infected, and they have some peripheral nerve damage. Whereas people with lepromatous leprosy have huge amounts of growth of those microorganisms. There's microorganisms growing everywhere in their macrophages. These people can spread leprosy to others really easily. And these people have disseminated infection. So they have infection throughout their whole body. So they are going to end up having damage to bones, damage to cartilage, and nerve damage throughout their body. They are also going to have really high levels of antibody responses. So they'll have hypergammaglobulinemia. You could imagine that if you got infected with the organism, Mycobacterium leprae, you would prefer one outcome to the other. And so we might wonder, well, what dictates which of these two outcomes an individual has? And when we actually study these patients in detail, we find that those patients who have tuberculoid leprosy are making Th1 cytokines and are making a Th1 response to this organism, while patients who have lepromatous leprosy are making a Th2 response, Th2 cytokines in response to this pathogen. And so the difference in whether you make a Th1 response or a Th2 response to any given pathogen can give a big difference in the outcome of your infection and what type of pathology you see. The reasons for this can vary quite a bit. One of them can be due to genetics. You might be genetically predisposed towards making more Th1 responses or making more Th2 responses based on certain polymorphisms you have in cytokine genes. There are actually some pretty famous polymorphisms in two of the strains of mice that immunologists usually use. There's one mouse that kind of always makes Th1, and there's one mouse that always makes Th2 because of genetic polymorphisms. There are also differences based on age and differences based on a number of other factors that can bias you towards a Th1 or a Th2 response. We can also look at infection with another microorganism, Leishmania. And first, if we look at the yellow mouse is a Balb C mouse, and that Balb C mouse can be infected with this parasite, Leishmania. When we infect our Balb C mouse with the Leishmania parasite, those mice all die within about 70 days, 75 days. We can also see that the thing that kills these mice is actually their Th2 response. It's the fact that these mice happen to make a Th2 response to Leishmania. So when we infect our mice with Leishmania, we also give them an anti-IL-4 antibody, so we block IL-4 signaling in these mice, then the mice live happily ever after following their Leishmania infection. So here in this case, the Th2 response is causing a pathologic or pathogenic response, which we don't see coming from the Th1. And we can see this with lots of different types of infections. So for example, if we have a T cell that makes a response to a helminth, so some type of parasitic worm, if that response is a Th2 response, we get the production of the cytokines that are shown on the left side here, and we do things like, increase number of goblet cells, make more mucus, make more eosinophils, have IgE, degranulate mast cells, and all of those things together are protective. And so we get better after our helminth infection. Yay! If, however, we make a Th1 response to that same helminth infection, now we're going to turn on macrophages, and actually the macrophage helps the parasite, not us, so that's going to make our life worse. And we're going to make IgG, which is not effective against parasites, so that's going to make our life worse. And in fact, this response will lead to host damage or immunopathology. And so the specific type of CD4 response that is made will vary and will have a lot to do with our specific type of, our specific outcome. So I'm going to switch the order of the next couple of slides because I think they'll make a little bit more sense this way. So Th2 cells are often talked about in response to helminth infection. These are some examples of some helminths that cause important infections. There are roundworms, flukes, and tapeworms, or nematodes, trematodes, and cestodes, which can include a number of different really important diseases. Ascariasis is one I showed you at the beginning of the semester with the person who had hands full of worms. It's it's not good. We can also see things like guinea worm disease. We can see elephantiasis. We can see river blindness. All of this, thank you, Dr. Campbell, in terms of development of medications. And typically, the Th2 response is the immune response that's going to get rid of that helminth infection. So we typically will have a mast cell full of granules. That mast cell will be triggered when it has IgE on its surface that's cross-linked, and will start to produce huge amounts of toxic molecules to kill this parasite. And this is actually a parasite larva. This isn't even a full parasite with eosinophils surrounding it. So you can see the only thing those eosinophils are going to be able to do is shoot bombs at that larva to try to kill it. So we're going to see a lot of attempts to kill that parasite. But most of the time when I think about Th2 responses and the times when I suffer from Th2 responses, I think about having asthma and every other kind of allergy in the world. And so in the case of asthmatic patients, Th2 responses are in fact causing the pathology. We're getting excess mucus secretion, which is good if you're trying to flush something out of your GI tract, but is not good if you're trying to breathe, as well as a number of other big issues. And we can show that Th2 responses are really important here because we can look at animals who have kind of normal lungs and normal airways, and we can also look at animals that are missing Tbat. If an animal is missing Tbat, it makes no Th1 response. Because that animal makes no Th1 response, it also makes excess Th2 responses. These two responses are in balance with one another. So we take a mouse with no Tbat, that means it has excess Th2 responses, and what you can see is that the airway is inflamed. It has way more cells in it than the normal airway because all sorts of white blood cells have trafficked to that area. And you can see that we have collagen deposition around the airway. So the airway is less elastic and less able to change. And that airway can eventually come to the point of looking like this, where it's full of mucus. Now, just so you know, this part is where the breathing happens. So if you notice, there's not really going to be so much of the breathing in this lung because there's no place for the air to pass through. And so all of those can be the response of a particularly large amount of a Th2 response. Yes, Molly?
Asthma is associated, increased incidence of asthma is associated with increased pollution and particulate matter in the air.
Uh-huh.
Okay.
So, Molly Molly correctly points out that there are some interesting epidemiology going on here. And we can see that in certain parts of the world, there are higher incidences of autoimmune diseases, and this also includes asthma in this description. And people have been wondering for a long period of time, why is there this increased incidence of autoimmune diseases, allergies, asthma in Western countries, as opposed to in other parts of the world? One observation that you can make is the observation that is shown here. On the left, you can see that incidence of autoimmune disorders, allergy, and asthma, highest in red. And on the right, you can see the incidence of helminth infestation. So where people are getting helminth infections, highest levels in red. And what do you notice when looking at these data? Yeah, Malik. They're the opposite of each other. So it seems like there are high levels of allergy and autoimmunity in places where there are low levels of helminth. And this has led people to hypothesize something called the hygiene hypothesis, where perhaps exposure to helminths and production of a normal Th2 response protects you from making pathologic Th2 responses. So you can kind of imagine the idea that your body is going to have to make a Th2 response to something. There's going to be some need for balance in your immune response. There has to be a Th2 response. If you never see a Th2 pathogen in your life, then your body is going to make that Th2 response against something inappropriate. Whereas if you actually do see Th2 pathogens, you're going to have a normally balanced Th1 and Th2 set of immune responses. There's a lot that's understood about this hypothesis epidemiologically. Relatively little is understood in terms of mechanism. So the idea is that in childhood, the development of the immune system is held back if you do not have exposure to all different types of pathogens, thus meaning that it becomes poorly educated, exercised, and inexperienced, and might start fighting imaginary infections. And if we just look simply at the epidemiology of this, at people who statistically are more or less likely to have asthma and allergy diseases, we can actually see a number of ways that this seems to play out. So individuals from westernized countries tend to have more allergy and asthma. Individuals from smaller families have more allergy and asthma. And in fact, it's not listed here, but actually being the oldest child makes you more likely to have asthma than being the younger child. And what you could kind of imagine is like when you're the older child, your parents keep your house all nice and clean, and then when you're the younger child, they're like, whatever. And because you have other children around, that's going to increase the microbial burden in the house. So the affluent urban homes, those with high antibiotic use, those with good sanitation, all may be associated with those higher levels of allergic disease. You could also imagine, Molly, that might be a pollution effect based on inflammation of the respiratory tract, sort of further pushing this. And so things like whether you're an only child can push you towards having asthma, whereas going to daycare makes you less likely to have asthma because you're around other dirty little kids. Growing up on a farm makes you less likely to have asthma because you are around you're around microbes and things like that. I will admit that I look at this and I'm like, thanks, Mom and Dad. More reasons. It's not just your genetics. You also had to give me all of these allergies for these reasons too. But also when in grad school, whenever any of us would sneeze, somebody would look at them and be like, your parents clearly didn't let you eat enough dirt as a child. There are actually some really interesting hypotheses out there about how to sort of deal with some of these things. There are people who propose that the way we should be able to get rid of and cure a lot of allergic diseases is to expose people to more helminth antigens. If we were to rebalance responses. And so some people have proposed feeding people dead schistosome eggs, like in yogurt, in order to push your immune system back and induce a physiologic Th2 response. There's one other explanation that others have made for the hygiene hypothesis, and the idea here is that in a population, we have some variation in general strength of the immune system. If some portion of our population is infected with parasites, if most of our population is infected with parasites, then all of us are going to have our immune system kind of pushed lower. And so the immune system is going to be kind of pushed down a bit, and very few people are going to have a super high immune response that leads them to some kind of pathology like autoimmune disease. But if you live in a clean environment, you have nothing pushing your immune system down, and so a higher proportion of people are going to have that high immune response that's over the threshold for allergy. So the balance between Th1 and Th2 responses may not be the only explanation for these data, these epidemiologic data, but you will very frequently see this invoked for details of allergy. And this is where all of those ideas that you've heard of about how, you know, real infection makes your immune system stronger and all that stuff, all of that is actually kind of like taking this a weird way. The idea of trying to make your immune system stronger with real infection. All of it is officially coming from this, and we don't have mechanism behind it. We know that people who are on farms get fewer allergies. Yeah, Emilio.
It's related to the thing that you said about feeding people eggs in an attempt to restore the balance.
Mm-hmm.
Would that work as an adult? Could you restore the balance as an adult, or is this more of a preventative measure as to do it to children?
There have been trials of it in adults that seem to have worked. And if you ask me how I feel about that, say, I don't know, like April 1st-ish, how I feel about doing something like that to get rid of allergies, I'll be like, give me the schistosomes. Because I have real bad allergies. So for a long time, these two subsets were the only two subsets that were known. I'm going to go slightly out of order in that the next one that was described is not the next one I'm going to do. I'm going to come back and do that one last. So technically, the next one that I talk about was the fourth one described. Sadly, for all of us, it is not called Th4. Instead, it's called Th17. And you might look at that and be like, oh my gosh, they really don't know how to count. But you're going to like the fact that it's named Th17. So Th17 cells were described around 2007. Their responses are polarized by a combination of cytokines. And so this is key. They have a combination of TGF-beta plus either IL-6 or IL-13, or sorry, 23. So the combination part is really important here. Th17 cells are polarized by a transcription factor named ROR gamma, sometimes ROR gamma T. And Th17 cells got their name because they make the cytokine IL-17. So in most of the newer types of helper T cells, we've named them Th whatever number, and the number is the cytokine they make. So the ones I'm not going to talk about, Th9 and Th22, make IL-9 and IL-22. IL-17 seems to be really good for activating neutrophils. And this seems to be very good for extracellular bacteria and also fungal infections. Th17 cells took a while to understand. And if you read old literature, you might find slightly contradictory things. And let me give you one reason why you might find that contradictory thing. In the beginning, when people were doing their Th1 experiments, they would often do experiments where they had removed IL-12 from their mouse, or removed IL-12 receptor. There were different ways of doing it. Turns out that IL-12 has multiple parts, as does the IL-12 receptor, some of which are shared with IL-23. So there were some diseases that people said, this disease is caused by Th1 cells. And then when they redid their experiments in a slightly more careful way, they're like, this disease was caused by Th17 cells. And so there are a few diseases where if you look in the literature, there's this weird confusion between whether they're Th1 and Th2 diseases. Because in all of these, with all of these T-cell subset cases, if they go wrong, there's some disease state that can occur. Th1s, when they go wrong, lead to certain types of autoimmune diseases. Th2, when it goes wrong, leads to allergy and asthma. Th17 can lead to a number of different inflammatory disorders. There's some really nice data on Th17s and multiple sclerosis. And so Th17 is now really this sort of inflammatory kind of set of cells that turns off fungal or that fights against fungal infection. Another of the sort of new-ish types of helper T-cell subsets that are really well accepted are the TFH cells. TFH stands for T follicular helper. So one thing you might know about them is that they definitely do some helping. And there's something going on with follicles. And the job of these cells is to help B cells in the follicles. If you remember way back when, we talked about B cell development, and we got our B cell to the point where it left the bone marrow. And we never did all this discussion of B cells in the periphery. The reason why we never did B cells in the periphery is because in the periphery, our B cells require a T cell in order to help them out. And specifically, they require a T follicular helper cell. So we actually can't talk about the B cells and their peripheral functions until we knew T follicular helper cells. We needed their job as well. That's why we're going to do them on Wednesday. T follicular helper cells are polarized by the cytokines IL-6 and IL-21. They make the transcription factor BCL6, and they produce cytokines like IL-4 and IL-21. What I will say for T follicular helpers is that this is an extreme understatement in the number of cytokines that they are both polarized by and produce, and I am specifically giving you the ones your textbook does, though, so as to not get crazy confusing. I am not sure that I would say that those would be the ones I would pick for T follicular helper cells, but I am sticking with your textbook. Yeah, Malik.
If the TFH cells
Mm-hmm.
produce IL-4,
Mm-hmm.
does that affect TH2?
So it's not so much that they're they might affect TH2. They might sort of alter, especially antibody production, and push towards a somewhat more TH2 response. It is possible. Again, I am going with what your textbook says for cytokines here. These are not the cytokines I would have gone with off the top of my head. But I'm going with what the textbook says to avoid confusion. So these are sort of 4 really big types of helper T cells. And the final type that I want to tell you about is the one that was actually discovered third. So once upon a time, they were called Th3 cells, but now they're not anymore. Instead, these cells are called regulatory T cells, or Tregs. And I want to tell you a little bit about Tregs and their important features, and then I want to tell you some stories about them. So Tregs can be induced by TGF-beta. What you will notice here is that the TGF-beta is by itself and is not in combination with something else like IL-6 or IL-23. They say IL-2. IL-2 is for all T cells. That's wrong. IL-2 is for everything. There's nothing special about IL-2 here. So they get just TGF-beta. They turn on a transcription factor called FOXP3, and they make cytokines like TGF-beta again, and also IL-10. But the key thing to understand about regulatory T cells is their function. These T cells turn off responses of all other cell types. These are the off T cells that turn everybody else off. And so you can see that your book says inhibits inflammation. You can see that the book says inhibit anti-tumor responses. They basically are inhibiting all of the other types of responses. And you can see that here as well, that our Treg is inhibiting this neighboring cell. So these are really important in turning off responses. There is one word that's actually on here that I have not said throughout this whole process. And that word, remember how last time I told you that danger signals, when I said it, I was going to get struck by lightning? This is the other word that I'm going to get struck by lightning. That word is suppress. So just so you know, if you are ever writing about immunology or reading about immunology and you see the word regulate or regulatory, that means off or inhibit. We always use regulate or regulatory and inhibit. We never use the word suppress. And the reason why is that a long, long time ago, there actually had been described a type of T cell called a suppressor T-Cell. And everyone found this, everyone was all excited about the suppressor T-cell. It turned everything off. Suppressor T-cells were the coolest. And people did like crosses, hardcore genetics, to find proteins involved in regulatory, in suppressor T-cells. And they found this protein called IJ that was in the MHC class II locus, like IA and IE. They were super psyched. And then somebody sequenced the mouse genome, and there was no IJ. And so then everyone was like, no, suppressor T-cells weren't a thing. I wasn't into that. What are you talking about? And it was like people laughed at you if you said the word suppressor. And so no one ever says the word suppressor. I'm shocked that it's in these books. And so we always talk about how we inhibit or regulate other cells, and we never talk about suppression because like there's still some trauma in there. And the level of proof that was required to show that regulatory T cells were real was actually quite high. There is a very famous paper that first describes them. It's one that a lot of immunologists think is a sort of short list for Nobel Prize type of stuff, but we'll see. The other thing to know about regulatory T cells is that regulatory T cells can be made in two ways. So I have written one of those two ways up on the board here. And this is the way in which a naive T cell can be induced to become a regulatory T cell in the periphery. However, some T cells do not, or some regulatory T cells do not get made this way. Some regulatory T cells become regulatory T cells in the thymus. So way back when we talked about the thymus, I told you that there were 4 possible outcomes for our T cells. I told you that those that got low or no signal died of neglect. They failed positive selection. I told you that those that got a low intermediate signal were positively selected and became T cells. And I told you the ones that got a very high signal were negatively selected and died. And I said also there's something that happens when they get high intermediate. And that's all I'm going to say. And in fact, if we have these cells that have kind of, so here it's low, high, intermediate, and it's not showing you 0. If we have cells that don't have quite high enough affinity to be negatively selected, but are still a little iffy, are still kind of on the dangerous side, we induce those cells to become regulatory T cells. That process still happens by turning on the transcription factor, FOXP3. And they're still suppressive cells, but they never had to see antigen in the periphery to make that happen. This is that version of that figure from your textbook. And so here you can see all 4 outcomes from the thymus. When I showed you this before, I put a white box over this one, so you didn't see it. But what you can see now is that that low set of signals leads to death by neglect. The more intermediate or sort of lower intermediate signals are the cells that survive and are positively selected. The high cells become Tregs, and the very high cells become negatively selected. The Tregs seem to be about 5 to 10% of your CD4 cells, which overall is about 1% of your T cells. There are now arguments about regulatory CD8 cells. That's what I think about that. And B-regs, people are also starting to talk about. If you want to know about B-regs, we can call my sister, because she knows about that, and I don't so much. Regulatory T cells are able to regulate other cells in a number of different ways. Sometimes they can do some cytotoxicity, though I'm not going to focus on that a lot. But these cells also can inhibit APCs, so the APCs cannot turn on other cells. These cells can produce inhibitory cytokines like TGF-beta and IL-10 that can turn on other cells. There also seems to be a cell surface protein where these cells are able to inhibit other cells by contact, although that's less well worked out. And the first thing that these cells were shown to do actually had to do with cytokines. When Sakaguchi first did one of the seminal papers describing T regulatory cells, the protein that he found on the surface of T regulatory cells that was really high was CD25. CD25 is the IL-2 receptor alpha chain. And so this was like super confusing for flow cytometry for a long time, because that's on activated T cells and Tregs, so that was confusing. Then we found FOXP3 and everything got cleared up. But it's possible that regulatory T cells also can inhibit other T cells simply by stealing all the IL-2. Having the highest affinity for IL-2, basically binding all the IL-2, which deprives other T cells of that IL-2. Sometimes this is referred to as being a cytokine sink. And so these cells can also act as a potential cytokine sink to acquire all of that IL-2. And overall, all of our T cell subsets are able to cross-inhibit one another. So Tregs can inhibit each of the other cell types, and just like development of Th1s turns off development of Th2s, development of Th1s also turns off development of Th17s and T follicular helpers and so on. So we actually have full cross-inhibition between all of these subsets. One of the really important types of cross-inhibition is the one that happens between Th17 and regulatory T cells. We can see that in both cases, TGF-beta is produced. If TGF-beta is produced along with one of these cytokines, IL-6 or IL-23, remember, IL-6 was one that we saw like the first week of class as an inflammatory cytokine that's made when inflammation occurs. If there's TGF-beta plus inflammation, then the cell's like, oh, better make a response. Something bad's going on, and we push that cell towards Th17. Whereas if this inflammation component is missing, and we just have TGF-beta, then we can push that cell towards a Treg and be more tolerogenic.